you very much, Rafe. It's a great pleasure for me to open the meeting telling you about how sodium channels work as an electrical signaling machine. Now, someone has the clicker, which is also the pointer. Thank you. So we'll take a bit of a deep dive and look at how sodium channels work in detail at sort of the atomic level. But I think you'll see when we think of the sodium channel as a signaling machine, you can actually understand how it works. So to begin at the beginning, there it is. So in this classic experiment from the 1930s, the horseshoe crab eye was stimulated with different intensities of light, 1 or 10 times or 100 times the intensity. And the electrical signals, the action potentials that were generated in the eye were recorded here. And you can see that as the eye is stimulated with increasing intensity, there are more electrical signals. They come at higher frequency and the stimulus lasts longer. So this is just an early example of what goes on every time we think about anything in the brain. The nerve cells generate electrical signals, they communicate with each other, and the pattern and intensity of the electrical signals communicates the information from nerve cell to nerve cell. Now, it's perhaps a surprise that the brain makes electrical signals. There's no battery, no wires. How does it work? Well, the brain makes electrical signals by moving charged ions, sodium ions and potassium ions, back and forth across the membrane of the nerve. So these charged atoms, sodium ions and potassium ions, are like the electrons in your wires at home in your electrical appliances. So here is a nerve axon, a nerve fiber, and outside of it, sodium, with its chemical signal, Na, you'll see that again, is high on the outside and low on the inside of it. And the opposite for potassium. Potassium's high on the inside and low on the outside, and potassium goes by its chemical signal, K, symbol K. To make an electrical signal, sodium channels in the surface of the fiber open, and sodium carrying its positive charge moves into the fiber through the sodium channels, as I'll show you, and begins the rising phase of the action potential. And then very briefly after that, in milliseconds, potassium channels also in the surface of the cell membrane open, and potassium ions carrying their positive charge move out. They bring the electrical signal back to the baseline. So each time there's an electrical signal in a nerve, a sodium channel starts it and a potassium channel ends it, and the electricity is carried by the sodium and potassium ions. So we've worked on sodium channels for a long time. This is an early slide, cartoon slide from the 1980s, when we were able to isolate the sodium channel molecule from all the other tens of thousands of molecules in the brain. And we found that the sodium channel, and actually this is an SCN2A sodium channel primarily in these experiments, has a large pore-forming alpha subunit, as we call it. So the protein has 3 components called subunits. The alpha subunit, large pore-forming subunit, and then 2 smaller beta subunits. So the alpha subunit does all the work of the sodium channel. The beta subunits are good for getting the sodium channel in the right place and keeping it there. But we'll be talking mainly about this large pore-forming alpha subunit today, and most of the mutations that cause disease are in this particular protein molecule. Now, I've described this as a large protein, but it's actually a pretty small thing. Its size is 10 nanometers. What does that mean? 10 billionths of a meter. So think about how small that is. And it makes a small electrical signal, as you might guess. It makes an electrical signal recorded here for one single purified sodium channel. It makes an electrical signal of about 15 picoamperes. And again, what does that mean? That's 1 trillionth of the 15 amperes you have in your household circuits. So it's a small molecule. It makes a very small electrical signal, and your brain works on that entirely. So proteins, like the sodium channel, are made up of long series of amino acids, building blocks. So sodium channel, a mammalian sodium channel, a human sodium channel has about 2,000 amino acids here. And they're organized in a long string. And then the sodium channel folds that string up just like a ball of string and makes a three-dimensional picture from it, a three-dimensional object. So looking at this sequence of 2,000 amino acids, we can see that it folds back and forth. It's threaded through the membrane 24 times, as you can see in this diagram. And it has a repeating structure. Each 6 transmembrane segments make a domain, domains 1, 2, 3, and 4. You'll hear this often during the day, I think. In each domain, there are 2 functional modules. And key point here, a voltage sensing module, which tells the sodium channel what the membrane voltage is across the plasma membrane. And that's made up by the 1, 2, 3, and 4 transmembrane segments, or S1 through S4. And then, of course, there's a pore through which the sodium ions move, and that's made up by the S5 segment and this little bit of protein in between that we often call a P-loop. This is one domain of the sodium channel structure. It's repeated 4 times. So these domains are not identical to each other, but they're similar to each other. After the sodium channel opens, it undergoes a process called inactivation. We'll see in detail how all this works. And this region here that connects domain 3 and domain 4 is important for the inactivation process. Now, in the last decade in our lab, we focused on trying to understand the sodium channel in its atomic detail. And in order to do that, we've taken advantage of the discovery of this little sodium channel over here. This is a sodium channel that David Clapham and his colleagues discovered in bacteria. Who knew? Bacteria don't have nerves. Why on earth do they have a sodium channel? So they have an ancestral sodium channel whose structure is just exactly like one domain of an SCN2A channel or other mammalian sodium channel. And this channel works by getting 4 of those identical domains together to form the channel, instead of having 4 that are similar to each other and are linked. So we use this as our structural model, and it's surprisingly similar to this core part of the mammalian sodium channels. So we use a technique called X-ray crystallography, and I won't try and tell you about that except to say that one uses X-rays to see these small molecules. And here's an image from X-ray crystallography of that bacterial sodium channel. On the left, looking down, in the center here is the hole through which sodium ions move. And here you can see the amino acid side chains that actually select sodium and let it go through and prevent other molecules from following it. This blue region is the pore forming module that I mentioned on the last slide. And it is surrounded by 4 voltage sensing modules that are in these green and red colors here. And on the right side of the slide, you can see the pore forming module now looking at it from its side, and it is again in blue. And you can see each voltage sensing module around the periphery. Each one is composed of 4 transmembrane segments, S1 through S4, and again, colored green. And now, crucially, in this image, you can see for the first time the linker that connects the 2 modules. This is in red here. It's called the S4, S5 linker because it connects segments S4 and S5. Mutations in voltage sensors, in the pore, and in the S4, S5 linker, all are present in SCN2A disease. Now, in the next slide, I'm going to show an image which is just one voltage sensor and the pore module so that we can focus on how the voltage sensor works. In some ways, this is the most unique aspect of the sodium channel because it senses the change in the membrane potential and it changes shape and it causes the pore to open and let the sodium go through. And we've just reached a new level of understanding of this from our work. This paper here, published last week, 7 days ago in Cell, shows the structure of the sodium channel in its resting state. And we already knew the structure of the sodium channel in its activated state. So now we can make a movie, and this is a movie, of how the sodium channel works when it goes from being resting to being activated. And I'll tell you the story and then we'll run the movie. So I want to focus on this voltage sensor, and I'm just showing one of them here. And now a new feature, the key thing about voltage sensors is they have positively charged amino acid residues in them. They're blue. 1, 2, 3, 4. See the 4 blue amino acids there. Notice that 3 of them, 1, 2, 3, are on the lower side, that's the intracellular side, of this key marker here. We call this the hydrophobic constriction site. Don't try to remember that. It's sort of the rubber seal that seals the channel. So 3 are inside. When I run this little movie in a minute, this S4 segment will move out. These positively charged residues will move out. They will pull on this S4, S5 linker, and they will pull open the intracellular end of the pore, which is right here. And that's how the sodium channel opens up. Now, it's a bit of an experiment. I don't know if I can run the movie from here. Do you know if I can run it with the clicker, or do you want to start it? Ah, there it goes. All right, good. Okay, so see what's happening? These positively charged amino acid residues are moving out. They are pulling here, and they are opening there. So this is how the sodium channel works. In the resting state, the nerve cell has its resting membrane potential. It's negative inside. That negative potential is actually pulling those positive charges in and keeping them there until the cell is stimulated. Then when the cell is stimulated and the membrane potential is depolarized, it releases that force, that electrical force that's pulling these gating charges inward, and they move out. One more time. And so you see the whole cycle there. Outward movement of the gating charges, pulling on the linker, opening the pore. So see if I can get the next slide here. There we go. So you've seen the left part of the slide before. What I want to point out to you now is this pore region. Of course, it's very important. It's how the sodium gets through and how the channel prevents other ions from moving in the same way. So on the right, in the gray area, is shown an image of the water-filled space in the center of the sodium channel through which the sodium ions move. So they come first to an extracellular funnel, then they come to a narrow spot, the ion selectivity filter. Crucial, as the name indicates, it selects sodium and tells everybody else to go away. Sodium ions go through into this central cavity. It's a large space in the sodium channel. And then the second very important thing that I want you to remember, intracellular end, there's an activation gate. These 2 transmembrane segments, they're called S6 segments, come together with 2 others that aren't illustrated in the slide for clarity, to form a tight gate here. No sodium ions can pass when the activation gate is closed. So that's the resting state. As you saw in the last slide, the resting state opens when you go to the activated channel, and you'll see that better on a subsequent slide in just a minute. Okay, so we want to focus on two things: the ion selectivity filter, the narrow spot, and the activation gate. So this is another movie from the paper we published last week. Now you're looking as if you were sitting inside the nerve cell and you're looking out. Here's the voltage sensor. You can recognize the blue gating charges here, but now they're going to move away from you into the slide or into the backboard. And you can see the intracellular end of the pore right here. It's tightly closed. You can see these little amino acid side chains. There's no space for sodium ion there in the closed state. And you can see this really crucial linker, the S4, S5 linker that's highlighted in blue. So when we run this movie, the voltage gating charges will move out like that, as they did before, but now out is into the blackboard, and you're looking at the bottom of the channel. You can see better the pulling motion on the S4, S5 linker here. It really makes a big movement for a molecular size thing. And crucially, the pore is opened right there. So closed, open, closed, open. So that's what lets the sodium come through. And the opening is driven by the changes in voltage, which makes this an electrical signaling machine. So let's carry on here. So once the sodium channel is open, sodium comes in through the ion selectivity filter. Let's look at that in a little more detail. So here it is right here. You've seen this slide before. Now if we just flip it and look as the sodium ion would see it coming from outside the cell, the ion selectivity filter is a roughly square array of amino acids that have negative charges on them. So they attract the positively charged sodium ion. Sodium ions in solution go around with a coating of water. They strip off some of that water, and in a very specific chemical way, they let the sodium go through, and they reject calcium, potassium, and the other kinds of ions that are in the extracellular fluid. Now, if we look a little more closely at this ion selectivity filter, we see it actually has three parts. Now we're looking at the side, and what you saw in the last slide was just this part here, the outer edge of the ion selectivity filter, where there are negatively charged amino acid residues. The sodium ions then interact with two additional way stations as they go through here, a central site and an inner site. So there are really 3 points of contact and selectivity for sodium as it goes through the selectivity filter. Again, all designed to make this sodium specific. On the right, we discovered, using a technique called molecular dynamics, that the negative side chains, which are shown here, and the negative charges in red, interact with sodium and actually move with it. So they kind of chaperone or catalyze the sodium movement through the selectivity filter. And this is another important aspect of the selectivity. This does not occur for calcium or potassium ions. So sodium channels can't be on all the time. You turn them on and then they have to inactivate so that you can make another electrical signal. As you saw, the information is in the frequency of many electrical signals. So just opening the sodium channel isn't enough. You got to close it down. It has two inactivation processes to do this. First one's shown on this slide. In this case, slow inactivation. Sorry, we've messed up the label here. A little Mac to Mac translation problem. Slow inactivation happens through an asymmetric collapse of the pore. And I've taken two sections through the pore here to show you how it works. So here at the selectivity filter, which used to be square, in the inactivated state, it's more like a rectangle or a parallelogram, and this is poorly conductive. It doesn't fit the hydrated sodium ion very well. And then another snapshot, again at the activation gate, another place that we've talked about. Normally, it is symmetrical. You see this is a circle that the ion goes through, but in slow inactivation, two of these S6 segments come close together, and they block the exit and essentially make it so the sodium channel is no longer conductive in this slow inactivated state. On the bottom, we're looking just right here. It's like a snapshot, but you're looking up at it, like from inside the cell. Closed, open, 10.5 angstroms, for those of you who speak angstroms, and inactivated. So closed, symmetrical, open, symmetrical, inactivated, not any longer symmetrical, more like a rectangle. So mammalian sodium channels also have a new invention of fast inactivation, and showing that on this next slide. They use this little linker right here that's connecting domains 3 and 4. Now, this part of the protein isn't present in the bacterial channel. They hadn't invented it back in bacteria days. But all of the vertebrate sodium channels have this structure, and this acts as an inactivation gate in a very simple way. It just folds in and blocks the pore. And that's shown on this next slide. Didn't go forward. Let's try it once more. There it is. So this is another movie. So here's the inactivation gate of an NAB 1.2 or SCN2A channel. We determined its structure some years ago, fastened onto our bacterial channel model. And now if you can run that movie. Have some sodium ions out here. You knew sodium was green, right? All right, so here's the sodium. The voltage sensor moves. All right, voltage sensor moves. Pore opens, sodium comes through. And then for a brain channel, within 1 millisecond, that's 1/1000th of a second, the inactivation gate closes. It makes sure that electrical signal is always brief, turns it off, and is ready for the next one, as you saw in that chain of electrical signals in the example from the horseshoe crab at the beginning of the talk. So every time a brain neuron talks to its neighbor, the sodium channel's going through this whole cycle of molecular events within a millisecond and making the electrical signal that tells the neighboring neuron what's going on, what the information is, and what it should do. Okay, just one more topic briefly. So sodium channels are a target for classical drugs. The one that you're most familiar with, you've taken often when you go to the dentist, and the dentist numbs your tooth with an injection. It's injecting a sodium channel blocking drug, often a drug called lidocaine that we'll see in a minute. And what does it do? Well, it blocks the sodium channels so that your nerves in your mouth can't make an electrical signal and tell the brain about the bad things the dentist is doing to you. So that's a really important drug, and it's very widely used, as you can guess. Okay, so how do those drugs work? Well, here's a cartoon from the 1970s. That's a long time ago. Turns out the cartoon is right on the mark from my colleague Bertil Hille at the University of Washington. Bertil, based on physiological studies, proposed that these drugs go into the pore and they block it. They sit just under the ion selectivity filter and physically block the pore. And boy, was that a good model, because that's exactly what happens. And we can show that now that we have structures. We can put drugs in and see where they bind. So here's cartoon version. So the amino acid residues that make the local anesthetic binding site are the colored balls here, right under the selectivity filter. So the drug blocks there, sodium can't go through. Here you see them looking from the top. Again, if you were a sodium ion and there was a local anesthetic molecule like lidocaine bound just there, well, you'd be stuck. You couldn't go through. And so that's how these drugs work. They block the pore. Now, recently, we've been able to actually image the lidocaine in its binding site in a sodium channel. And that's shown on this slide. It's a little bit difficult to read, so I'll lead you through it. For those of you who read organic chemistry, there are the drug molecules, benzocaine, lidocaine, the most common of the local anesthetics, and flecainide, a different one. Here's a drawing of the sodium channel. It's different from what you've seen before, but the selectivity filter, SF, is right here. The central cavity is right there, and the drug binding site is right here. And how do we know that? Because we can see the electron density with X-rays or electrons and show that this is lidocaine. It's right there. The green mesh is the electron density. That's the measured that value of the electron density. and it fits the lidocaine molecule exactly. There is the drawing of the lidocaine fitting exactly in the mesh. So no doubt about how these drugs work. They block the pore. They sit right under the selectivity filter. Here's a different drug called flecainide, used mainly in cardiac arrhythmias. And these drugs then are truly pore blockers. Okay, so, and last slide, which you've seen before. We'll just finish up with this one. Why don't you run this movie a little bit? I'll just make one more point about the drugs. So when these drugs work, they've got to get in here, right? They've got to get into their binding site, which is inside the pore. They're sort of big. So what happens is that when the channel opens, the drugs go in and they block. So it means the binding and block of the drugs is rather complicated because they can't just access their binding site anytime. They can only do that when the channel's open. That makes these drugs especially good for conditions like cardiac arrhythmia or epilepsy because the channels open more often in those conditions. And so you block the channels that are firing fast and you don't put the rest of the brain asleep or stop the heart. So that's a key aspect of these kinds of drugs. It's called use dependence. Okay, so this is our tour of the sodium channel at the atomic level. I hope that by treating the sodium channel as a machine, it helps you to understand how it works. And now the last very important slide, I won't go through, but it lists the many people over the years who've contributed to this work. Oh, last slide, I have to do that. There it is. People in my lab, the underlined ones who are still there and did the recent work, collaborators at University of Washington, collaborator at Toronto, and the NIH. Thank heaven for the NIH and grant money. And so I want to thank you for your attention. Next speakers are going to tell you about mutations, and I hope this background about how the channel works will help you see how those mutations are messing it up. Thanks.